Hello students, this will be a review of Chapter 3. And in Chapter 3 we're going to be talking about cellular structure and then proteins in terms of how cells make proteins and how they degrade them. So these are the topics that we'll be covering in this chapter. Again, the structure of the cell and then how the cell makes proteins, so protein synthesis and degradation. We're not going to really get into protein secretion. So first off, we'll talk about the two different kinds of cells. We have eukaryotes versus prokaryotes, and both are microscopic structures. The difference between them is whether they have a nucleus or not. So karyote means a nucleus. A prokaryote does not mean a cell that has a professional nucleus. It is actually a cell that was pre-nucleus. So it is a cell that does not have a nucleus. It is a very primitive kind of cell. As opposed to eukaryotes, and the term eu means new. Okay, so these newer cells, relatively more advanced cells, these have nuclei. So nucleated cells versus non-nucleated cells. Human cells, and those are the cells that we'll be talking about through the chapter, are of the eukaryotic type. Okay, so we do have nuclei, and we're more sophisticated than the prokaryotic cells. This right here is kind of like a schematic image of what a human cell looks like. Know that it is not a 2D, it's not a flat structure. It is you want to think of it maybe as a soccer ball. Okay, it is a 3D, it's a 3D structure surrounded by a cell membrane on the outside. And then on the inside, you have cytosol, which is the gelatinous material with all of these different organelles swimming and floating within that cytosol. So for instance, you have a mitochondria, you've got Golgi Apparatus, and so on. And we'll talk about these organelles throughout the chapter. Hey, a common mistake is that we tend to kind of use the term cytoplasm and cytosol interchangeably, although there is a difference. So the cytosol, which again is that gelatinous material, plus the organelles that are swimming in it, both of them together combined make the cytoplasm. So basically your cell is made out of a cell membrane on the outside, the nucleus that controls everything, and then the cytoplasm. Cytoplasm is made out of the cytosol plus these little tiny organelles. Another common mistake that students get into would be confusing organs with organelles. An organ is a macroscopic structure, meaning that you could see it with the naked eye. So, for example, brain, heart, individual muscles, these are organs. Organelle, on the other hand, are these microscopic, and again, you'll find them swimming within the cytosol of individual cells. Now, first off would be the cell membranes. Okay, the cell membrane or the plasma membrane is there to protect the cell. So one of the ways you can get rid or kill a cell would be by destroying the cell membrane. It keeps everything protects everything on the inside. It regulates what can go in and what can go out, and we call that selective permeability. So it won't let everything in, and it definitely won't let everything out. It's very selective on what things it will allow to pass through it. It can also detect chemical messengers. So it has like these receptors on the outside that have the ability to receive the messages coming from the surrounding cells. Okay, the cell membrane links different cells together, and we'll see, the different kinds of membrane junctions in a minute. And then the cell membrane also anchors the cell to the extracellular matrix, meaning that these cells are not just floating within our bodies. All of the cells, are linked together and are linked to the surrounding matrix. If you take a look at the structure of the membrane, and we talked about phospholipids in chapter 2. Remember we said that phospholipids are amphipathic molecules, meaning that they have one side that it likes water, and we call that the hydrophilic head, and it had the other side that did not like water, and we called that the hydrophobic tail. Well, if you put two layers of phospholipids together, that is what makes our cell membranes. So cell membranes are bilayers, two layers of phospholipids. I'm going to go to another picture so that you have a bigger image of what we're talking about. So here is one layer of phospholipids. These are the head, hydrophilic heads, and these are the hydrophobic tails. Well, this is another layer, again, your hydrophilic heads and your hydrophobic tails. And you see that the heads that like water are towards the areas that have water. So just to orient ourselves, this is the inside of the cell, intracellular fluid that has water. And this is the outside of the cell, extracellular fluid that also has water. So these hydrophilic heads are facing the sides that have water, while the hydrophobic tails are hiding on the inside of the cell membrane. Among, kind of like floating within that, these fatty acid hydrophobic tails are these darker yellow cholesterol molecules. Okay, and these kind of give like a little skeleton, so to speak, little support to the cell membrane. In our cell membranes, we have lots of proteins. So all of the, that green, purple, and blue, obviously our cells aren't color coded, but just to kind of illustrate what we're talking about, there are lots of proteins. Some of these proteins are known as integral proteins. They are integrated within the cell membrane, meaning that they are a part of the cell membrane. So if you remove these proteins, you have destroyed the cell membrane. While others are not integral proteins, okay, they're peripheral proteins. They are related to the cell membrane, but again, they are not an integral part of the membrane. So if they're removed or destroyed for whatever reason, the cell membrane will remain intact. So let's talk about the integral proteins. Okay, some of them act like channels. So you see that this is there's a tunnel that allows things in and out of the cell. The protein is also amphipathic. So you'll find that the polar regions, the hydrophilic regions, again, are towards the water areas, while the nonpolar or the hydrophobic area is going to be in the middle. Okay, these proteins can either be channels like this one. They could be receptors. So again, receiving information from surrounding cells. If they expand or if they span through the whole width of the cell membrane, we call them transmembranous proteins. Okay, so they go through the whole length of the cell membrane, kind of protruding on the inside and a little bit on the outside. If you compare that to a peripheral protein, so peripheries means kind of like on the sidelines, and that's what these peripheral proteins do. They kind of dock right by the cell membrane, and they are related to these transmembranous proteins. So a lot of the times, the integral protein or this transmembranous protein will receive information and that take it to this peripheral protein in order to take that message towards the inside of the cell. And we'll talk a lot more about this in detail when we get to cellular messaging and cellular communication. So how do the cells, what are the different kinds of membrane junctions? We've got desmosomes, tight junctions, and gap junctions. Know that all of these cells have different functions. So the ways they bind together are going to also be different depending on the function of the cell. So for example, we'll talk about desmosomes first. Now these desmosomes are technically act like Velcro. Okay, so here is one cell, this is the neighboring cell, and you can see that there is a gap in between. That gap is known as the extracellular space and is full of extracellular fluid. We also have these dense plaques that are found docking by the cell membranes on the inside, and you could find extensions like these orange spider webs made out of different proteins, and these proteins are called cadherins. These cadherins interact with each other just like Velcro would interact together. Anchoring it to the inside of each cell are other little tiny spider webs made out of keratin filaments. Now, having that kind of junction, you'll find those in areas where you need a lot of flexibility. So, for example, skin. Okay, so the skin cells have lots of desmosomes. The next kind of junction is known as a tight junction. And you see here that at the site of the tight junction, there is no extracellular space. It's kind of like you've sewn the two cells together. And you'll find these tight junctions in areas where you need to prevent leakage. So for example, in the urinary bladder. You can also find it in the intestine. Okay, so again, this kind of shows you that the structure of cells really reflects the function of the cell. The last but not least kind of junction we are going to be talking about are gap junctions. So gap junctions are basically tunnels that allow the two cytosols or the two cells to communicate directly with each other. So things are going to be able to pass freely from one cell to the neighboring cell. And freely depending on, the size of the substance. So you see here that these gap junctions are very tiny. It'll allow the smaller things to pass, like ions, like sodium, potassium, calcium, and so on. But the bigger proteins, these molecules are too big and they won't be able to pass through the gap junctions. So the cell won't be losing its protein, but it does allow for the communication of or the passage of these ions. You have these gap junctions in areas where you need fast communication between cells. So cardiac muscle fibers, which are the muscle fibers of the heart, have tons of these gap junctions so that when the heart contracts, it contracts as a pump. All of the muscle fibers will contract at the same time thanks to these gap junctions. So we'll talk again, about them. So this was, I prefer kind of explaining things using the images, and then we'll go through them in the slides to kind of reinforce what we just learned. So desmosomes, again, you'll find them in between two adjacent cells, and you are going to have that space in the middle. Okay, so the cell membranes are not touching each other. The areas where the Velcro or those proteins are going to be radiating out of are known as the dense plaques. On the outside, the Velcro are known as cadherin proteins, and those are going to extend into the extracellular space, while the fibers that are intracellular are known as made out of keratin. It might not be on the slide, but now we know what it is. And in this, you'll find desmosomes in areas, again, that you need kind of like that stretch and that flexibility. For example, the skin. For tight junctions, we find these where some areas do not have any extracellular space in between the cells. Again, it's like you've sewn the two cells together at certain spots. And tight junctions are going to make barriers that prevent the movement of things between cells. So again, you'll find them in places where you want to prevent any leakage for for example, the urinary bladder and in the gut. For gap junctions, these are protein channels that link the cytosols of two adjacent cells. And the small diameter of these channels is what really limits what can pass through them. So you'll find the smaller molecules, as I mentioned, sodium and potassium, are able to pass through while the bigger proteins remain inside of their cells. Gap junctions are there for fast communication between cells, for example, the heart, and that allows the heart to pump as a whole, as a unit. Okay, when we learn about the conduction of electricity in the heart and on nerves, we will learn a lot more about the roles of sodium and potassium, in those scenarios. Now we'll move on to the nucleus. And the nucleus is kind of like the big boss of the cell. It has all of the genetic material, and it has the information on how to make protein. So when we get to protein synthesis, we're going to have to talk about the nucleus again, because this is where the manual is. It has all the instructions on how to make a protein. All of the protein is coded. Okay, the instructions are coded, found on their DNA. And DNA tends to kind of be in this, massive threads of chromatin. Okay, kind of looks like a ball of string. But at the time when the cell is ready to divide, that chromatin condenses and then changes into what we know as that infamous X-shaped chromosomes. Okay, so the chromosomes are when we'll see chromosomes in the cell when the cell is about to divide, but when the cell is not dividing and it's performing its function, the DNA or the genes are really kind of make this network of threads that we know as chromatin. The nucleus, because the DNA is very valuable and important, there is an envelope surrounding the nucleus. And the nuclear envelope has two membranes, an outer and an inner membrane. But again, instructions have to reach the cytoplasm. So there are nuclear pores. There are little openings in the nuclear envelope so that there can be communications between the nucleus and the outside organelles in the cytosol. So we will see that RNA will be made inside of the nucleus, and then that RNA can squeeze itself through these nuclear pores in order to reach the cytosol. Okay. Inside of the nucleus, there is a little tiny structure known as the nucleolus, so kind of like the baby nucleus. Okay, and the nucleolus has is made out of lots of protein and lots of RNA. Okay, and we'll see, the nucleolus is really where the RNA is being made. So this is an image of the nucleus. You could see here the condensed nucleolus, again, made out of RNA and protein, and, this is where it has lots of RNA because this is really where the RNA is being made. And then you could see all of that chromatin. Again, when the cell divides, we'll have the X-shaped chromosomes. And then you could see the nuclear envelope that has two membranes, an outer membrane and an inner membrane. And then at certain areas, you could see these little tiny openings known as the nuclear pores. And again, this is how different messages are going to be taken out of the nucleus in order to reach the cytosol. Now, ribosomes. So ribosomes are your protein factories. Okay, and there are a couple of reflexes that we need to kind of develop as we go through this chapter. One of the first reflexes we want is that reflex whenever you hear the term ribosome, you automatically think of protein. Okay, so protein factories are your ribosomes. And ribosomes are made out of RNA and protein combined together. There are different kinds of ribosomes in the body. So we are going to introduce these different kinds throughout the chapter. There are 4 kinds that we are going to want to be familiar with by the end of this chapter. Okay, ribosomes, the interaction between the different kinds of ribosomes is how the cell makes protein. Okay, and I'm not really going to get into the details of protein synthesis because that is towards, the end of the chapter when we get there. But for now, let's know that ribosomes are your protein factories. Okay, ribosomes can either be free ribosomes, so they're just kind of free floating within the cytosol, and we call those free ribosomes, or ribosomes can be attached to the wall of the endoplasmic reticulum. And we'll talk about the endoplasmic reticulum, I believe, maybe in the next slide or two. And if it is attached to the endoplasmic reticulum, it gives the surface of the ER that rough appearance, and that is why we call it the rough ER. So I'll come back to these in a minute. I just want to show you the images of what an ER looks like. So ER stands for endoplasmic reticulum. Okay, and this is what an ER looks like. It is basically a maze of membranes. And this ER right here does not have any ribosomes on its surface. So the exterior is smooth, hence the name smooth ER. Smooth ER makes lipids. And a good way to remember this is that, lipids or fats are kind of like smooth in their texture. So smooth ERs are the organelles responsible for making lipids. And another function that smooth ERs do are the detoxification of drugs. And when we mention drugs in the course, I do not mean street drugs. I mean medications. So a lot of the medications that we take are going to end up being metabolized by the smooth ER in the liver. Okay, so for example, Tylenol, okay, which is, a very famous kind of drug that a lot of people take for pain or fever or so on. So Tylenol, or acetaminophen is the generic name, is going to be metabolized by the smooth ER of the liver. Okay, so all of the lots, or most, I should say, of the metabolism of different drugs occur in the liver. So liver cells will have tons of smooth ERs. On the other hand, you want to make sure that if somebody has, say, liver failure, so their smooth ERs in the liver is not as, it's either lacking in number or lacking in function. So you would have to adjust their doses according to the degree of failure. Okay, so something to kind of keep in mind as we're trying to tie, what we're learning with different clinical things. So again, smooth ER, number one, lipid synthesis. Second function is to metabolize and detoxify drugs and toxins. Third function, which is really important too, that it stores calcium. Okay, so when we talk about muscle contraction, calcium has a very important role in for muscles to contract. Calcium is going to automatically appear from its storage area, which is the smooth endoplasmic reticulum. Okay, so again, keep that in the back of your head somewhere, and we will come back to that when we talk about muscle contraction. Now, on the other hand, if ribosomes are found on the surface of the endoplasmic reticulum, giving it a rough appearance, we will call those ERs rough ERs. Rough ERs is where proteins are made. So we have the interaction between the ribosomes on the top and the ERs, where, the whole process, the protein will keep on going through this maze, and it'll get bigger as it goes through the ER. So, again, you guys can go ahead and read what we just talked about. I kind of prefer to use the images as opposed to reading the slides. Okay. Now for the Golgi apparatus. Again, this is a maze of flattened sacs, usually tied to the rough ER, where proteins are kind of made in your rough ER, and then they approach the Golgi apparatus where they are packaged. So you want to think of the Golgi apparatus kind of like the FedEx store, where you take your package there and they package it for you, and then they send it to the outside world. So that's what the Golgi apparatus does. It takes the protein from the rough ER, and sometimes it has to change its shape a little bit or add a little bit of carb to make it into a glycoprotein. Okay, and it won't send the protein out without being boxed in something. So it gives it a membrane, and then it secretes it out. And that membrane, that structure is known as a secretory vesicle. The membrane of this vesicle is actually derived from the wall of the Golgi apparatus. So the Golgi apparatus sacrifices a little bit of its membrane to surround the protein that was made and then send it out to the cytosol and then maybe secrete it out of the cell. For the mitochondria, and this is another one of those reflexes, you want, whenever you think of mitochondria, you automatically think of powerhouse of the cell. So all of this protein synthesis and making proteins and lipids and so on need lots of energy, and our energy, the energy that our cells use are known as ATP molecules or adenosine triphosphate, and it's in the mitochondria where that is made. The structure of the mitochondria is pretty simple. It has an outer mitochondrial membrane, an inner mitochondrial membrane that has all of these shelves, and these shelves are known as crista. And then there's a matrix on the inside. Okay, so kind of like a gelatinous material on the inside. These crista is where most of our ATP is being generated. Okay, depending on how much energy each cell needs, the number of mitochondria can vary. So I want to ask you guys a question and think about it. So which cell would need more mitochondria? Would it be your muscle cell or say a skin cell? Okay, so obviously your muscle cell will need more mitochondria. Which is true. Muscular cells have tons of mitochondria, while skin cells would have less mitochondria. A person who is athletic and trained, their skeletal muscles will have more mitochondria than, say, somebody who is not athletic. Okay, so again, the number of mitochondria really varies according to the kind of cell and depending on the level of training individuals have. Now, lysosomes are spherical shaped organelles, again, surrounded by a single membrane, full of enzymes, full of acid and digestive enzymes. Okay, just a disclaimer here. These enzymes are not to digest food. Okay, these digestive enzymes are to digest bacteria. So we expect our immune cells to have lots of lysosomes, which is definitely true. Okay, so our immune cells have tons of these lysosomes. They are, if you want to know the literal translation of lysosomes, lys means to break, so lysis, and some means body. Okay, so these little tiny bodies have acids and digestive enzymes that are going to look for bacteria and break them down, and also look for debris from dead cells. Okay, so a lot of our cells, as they age, they are going to die off, and our body does not want to keep these dead cells as trophies or for souvenirs. We have to get rid of those dead cells, and it is the function of these lysosomes to digest them and get rid of that debris. Okay. Now, peroxisomes are kind of the same thing. So they are also sacs that are made out of a single membrane, and they are also full of different enzymes. Peroxisomes, though, are responsible for getting rid of free radicals. So on a daily basis, our cells produce free radicals. And we talked about how free radicals can speeden up the aging process. So it's the function of peroxisomes to get rid of these free radicals, okay? And it also produces, in the process, it can produce hydrogen peroxide, which is H2O2. So yes, the hydrogen peroxide that we buy from drugstores, our body will take free radicals and change it into hydrogen peroxide, which is obviously a toxin. We cannot keep them. We can't keep H2O2 in our cells, but peroxisomes have enzymes to break that H2O2 down into products that are not, or at least less toxic. And that's where the name peroxisome comes from, because it has these enzymes that breaks down hydrogen peroxide. Okay. Now vaults. And vaults are the first kind of RNA that we are going to be introducing. vRNAs are just like any RNA, made out of a little bit of RNA and a little bit of protein. And this is a relatively newer kind of RNA that was discovered that covers the nuclear pores. Hence the name vaults. They're kind of like these vaults covering the nuclear pores. And the thought is, we really don't know what the function is exactly, but the hypothesis out there is that it regulates what can pass from the nuclei out to the cytosol. Okay, so it's kind of like a gate allowing or preventing certain things from going from the nucleus into the cytosol. And it is also thought that these vault RNAs are responsible for how our cells respond to different medications. So you'll find that a lot of, when we deal with patients or even, amongst ourselves, some people are totally good with taking Tylenol and they feel fine, while other people say, you know what, Tylenol doesn't do anything for me. I have to take Motrin, which is a non-steroidal anti-inflammatory drug, which is basically ibuprofen. And they respond to that. Okay, so is it that the differences in our bodies to respond to different kinds of medication, is it due to these vault RNAs? Well, again, that is the hypothesis. We are not sure yet. The cytoskeleton are protein filaments that are found within the cell to keep the cell structure. Okay, so we talked how about the cell membrane is basically 2 layers of lipids, of phospholipids. The cytosol is a gelatinous material. We have the organelles swimming. Well, what is going to keep that cell from collapsing? It will be the cytoskeleton or these protein filaments. We have different kinds of proteins depending on the kind of cell. So not each cell has all of these cytoskeletons. And the cytoskeletons that we will be discussing are actin filaments, also known as microfilaments, intermediate ones, and then microtubules. The first and the last one, so the actin and the microtubules, these are easily assembled and disassembled by the cell. So the cell can make actin filaments when needed and microtubules when needed. And then when really fast, and when they're not needed, it can. That is not the case though with these intermediate filaments. Intermediate filaments, once they're made, they're kind of more or less permanent, okay, and the cell has a harder time of disassembling them. So these are the structures of the different filaments. These are your actin filaments, and know that the filament is the whole chain, okay, made out of these little yellow beads, and each bead is known as a G-actin. So that is your protein subunit of a G-actin protein. G stands for globular, and you can see here coming from the fact that these look like little globes. The intermediate filaments, these filaments are made out of different kinds of proteins that we'll learn in a minute. And then microtubules, these are bigger, okay, in structure. They are cylindrical and hollow on the inside, okay, made out of a protein subunit known as tubulin, which is nice that microtubule, their building blocks are known as tubulin. So that helps us remember the name. So first one again is your actin filaments, and made out of the G-actin or the globular little proteins. Okay, these filaments help in the amoeboid-like movement of cells. So for example, immune cells, those have to, the white blood cells have to squeeze themselves in between other cells in order to reach where an infection is or where an inflammation is. So that amoeboid-like movement, kind of like moving like a caterpillar, is going to happen because of these actin filaments. We also use those during cell division and during muscle contraction. So when we talk about the little proteins that are found in a muscle cell, we are going to go back and mention actin and myosin and other proteins. So now we know that actin is one of the cytoskeletons, and that is important for muscle cell contraction. Intermediate filaments, okay, we said that they're made out of different proteins, and those, for example, keratin, desmin, and lamin. So if you remember, keratin and desmin comes from the word desmosomes. So remember, you'll find lots of these intermediate filaments in areas that need to be flexible and are exposed to a lot of mechanical stress, for example, in our skin. Now the microtubules, okay, made out of a protein known as tubulin, these microtubules are really important during cell division. Okay, so right here, this is like an electron it is an electron microscopic picture of mitosis, which means the splitting of the chromosomes or splitting of the nucleus. So in this area, we have what are known as centrosomes. And radiating from the centrosomes are these greenish microtubules that will pull the chromosomes apart here in the middle. Okay, so these microtubules help in the process of mitosis during cell division. Microtubules also make cilia. Cilia are, some cells have like kind of like eyelashes on the outside, and we will call these ciliated cells. You can find them, for example, in the trachea, which is their windpipe, in order to, and it'll make a wavy movement in order to prevent dirt and little tiny products or in little tiny particles from entering the lungs. This is a good time to take a break before we start protein synthesis. And it's also a good time to watch a video about protein synthesis so that when we discuss the whole process, you have a visual of how things work. And I am going to post the video on in Moodle. So welcome back, students. Hopefully at this time you have watched the protein synthesis video. Like I said, it'll make your life so much easier watching the video and having, that vision of how protein synthesis works before we talk about this in a 2D format. So the protein synthesis follows two different steps. Remember that the instructions are in the nucleus on your DNA. Okay, DNA cannot leave the nucleus, so it has to send a messenger. And that process of making a messenger RNA can leave the nucleus, is known as transcription. While the RNA will go into the cytosol, and we have to now translate the messenger RNA into a protein, and that process is known as translation. So you want to make sure that you know that the two steps and the order that they happen in. So transcription is first, translation is second. They are actually in alphabetical order. So if you look in a dictionary, transcription will be first and then followed by translation. So here kind of, shows you how the process works again. So the DNA has the instructions on how to make a protein, but again, it is trapped within the nucleus. So what happens is that the DNA unwinds and it makes a messenger RNA. That process again is known as transcription. Now the messenger RNA has the ability to squeeze itself through the nuclear pores, go to the ribosomes, and that's where the message will be translated into proteins. Now, whatever the function of the protein is, whether, it'll perform certain functions like contraction, or the protein is actually a hormone or an enzyme, it'll perform its function. So regardless really of what the function of the protein is, the process of making it is the same. This here kind of shows you, again, the part of the gene, in one of the strands of DNA. So remember, the DNA has to unwind, and we are going to be looking at these bases. So remember the bases, the adenine, thymine, cytosine, and guanine. We're going to read them in threes, and then each of them will make a messenger RNA. So there is an mRNA here in the middle. And then each one of them is going to stand for a different amino acid. So the DNA code, know that we have the four bases, the A, C, the A, T, C, and G on your DNA. So if you mix and match them, we can make up to 64 triplets. But the body only uses 20 amino acids. So we basically have 44 extra triplets that our body really doesn't need. So what happens is that a lot of these codes stand for the exact same amino acid. And that is what we know as redundancy. So I'll show you an example and we'll come back to this. Yeah, if you want to switch slides anytime here. There we go. So for instance, the UUC stands for the same amino acid. You can see here that all 4 of these code for the exact same amino acid. Same thing over here. Okay, so the fact that more than one code stands for the same amino acid is known as redundancy. So a lot of these codes are redundant, where, again, having more than one triplet standing for the same amino acid. Okay, and that comes from the fact that we have a combination, we have 64 possibilities of triplets, while the body only uses 20. And therefore, a lot of these codes can code for the exact same amino acid. Okay, one of the codes though is known as a start code. So the AUG code, and that's really the code, one of the codes that you want to know. So the AUG, which is on your messenger RNA, is going to indicate where protein synthesis should start. 3 triplets, 3 out of these 64, do not stand for any amino acids whatsoever, which means that this is where these are known as stop signals, where protein synthesis will end, since there is no amino acid that stands for them. So we could find them where it says stop. So these 3, UAA, UAG, and UGA, do not code for any amino acid in our bodies, which means that this is a signal to stop protein synthesis. The start signal is AUG. That stands for methionine. So know that all of our proteins, the very first amino acid in the sequence is always going to be methionine, because this is the start signal. Notice here that these are all U's. There are no T's in these codes. So all of these are found on your messenger RNA. Okay, messenger RNA is a long chain of bases. So how does the ribosome know where to start reading the code? It looks for the start code, or the start codon. And that's where the whole process of protein synthesis will start. It'll keep on reading the whole messenger RNA until it reaches one of these 3 stop codons, and that's when it stops making the protein and it releases it into the cytosol. So again, a lot of this will make a lot more sense if you've watched the video first. Okay. So how is it that this little strand of DNA that's inside of the nucleus, so this blue area represents the nucleus, this is your nuclear envelope, and the yellow area represents the cytoplasm. So how does the process work? Know that your DNA, again, is double helix, so we have to unwind it, break those hydrogen bonds between the bases, unwind the DNA, and expose one of the strands of the DNA. The DNA strand is made out of exons and introns. The exons have the instructions on how to make the protein. The introns, though, do not. So, when we actually transcribe this DNA to RNA, we will transcribe everything, exons and introns. So you see here, the darker ones are the exons and the lighter blue ones are your introns. this makes what is known as a primary RNA transcript. So it's kind of like when you're writing a paper, you write a transcript first, and then you go back and review it to make your final draft. That's what the body does in transcription. It'll first make a primary RNA transcript that has both exons and introns, and then, and this process happens through an enzyme known as RNA polymerase, enzyme, and then we have these little spliceosomes that are going to splice up the RNA and get rid of these unwanted introns. It'll stick the exons back together, and now we have made our messenger RNA. The messenger RNA exits through the nuclear pore and will go to a ribosome. Remember, your ribosomes is your protein factory. So the messenger RNA has to go to the factory in order to now translate that into a polypeptide chain. The translation is an interaction between messenger RNA, so there is right here your messenger RNA, and this bigger structure known as transfer RNA. Now, transfer RNA has a little area at the bottom that is a mirror image of these codes. So the code on your messenger RNA is known as a codon. The mirror image on the transfer RNA is known as the anticodon. And in order to remember that, the T in anti is found on your transfer RNA. So the transfer RNA will look for the complementary or the mirror image codon on the messenger RNA. So for example, UGG, its mirror image would be ACC. Now that we know that this is where this code stands for an amino acid known as tryptophan, now this transfer RNA will bring the tryptophan amino acid and attach it to the growing chain of protein. Know that all of this whole process is occurring at the ribosome. So that is your last kind of RNA we are going to be introducing, which is ribosomal RNA. And here you can see all three of them working together. So that is your messenger RNA, and we read again the codons in threes. The transfer RNA has the complementary bases or the mirror image bases, or known as the anticodon, and each one of these will stand or code for a specific amino acid. The whole process of protein synthesis occurs in this bigger yellow structure known as the ribosomal RNA, or the ribosomes. And you can see here that we read the messenger RNA. It's kind of like you're pulling the messenger RNA through that ribosome and reading from in this direction. The transfer RNA brings the amino acid. It'll attach it to this growing polypeptide chain and then leave the process. So you see here, this one is coming in, attaches it to the growing polypeptide chain, and then eventually leaves the process. And again, if you have not watched the video, I strongly recommend go to watch it, because this will make a lot more sense and it will be easier if you have watched the video first. Now, after the protein is made, again, we have made kind of like a transcript of the protein. Sometimes the proteins need to change a little bit, and that changing is known as post-translational splitting of the protein, where we can either keep the whole protein as is, or we can slice it into smaller proteins. So you see this big protein could be protein 1, or we can slice it into just the A portion, B portion, or C portion, or we can make it into these, bigger proteins like B and C together. Okay, so they're different proteins that can eventually come out of that one protein that was just made. So you want to think of it kind of like baking a cake. Okay, so you made this big cake. You can either give the whole cake to somebody, or you could cut it into smaller pieces, whether small or bigger pieces, and give that out to people. Okay, and that process of splitting the protein, the bigger protein, into smaller ones is known as post-translational splitting of a protein. So a lot of our proteins are made as bigger inactive proteins. And in order to activate them, we would have to split them to remove the part that is making it inactive, keeping the rest in the active form. For instance, insulin. Insulin is made by the pancreas. It's a protein hormone made by the pancreas. It is secreted in an inactive form known as proinsulin, which would be your bigger protein, proinsulin. And then that proinsulin is split to remove the part of the insulin that keeps it inactive, that's kind of locking the insulin. And then insulin itself is going to be released and now is active. Okay, so this whole process again happens a lot of times when we need to activate proteins. So these are the events again. So we kind of went through the images and so on. Here again is that little link to go to that video that I've mentioned a couple of times. So again, your first process is transcription, where under the control of the enzyme RNA polymerase. And if you break down the name, ASE means enzyme. And whenever you find the word ASE, that means you're talking about an enzyme. Polymer is a big molecule. RNA is RNA. Okay, so this enzyme is going to make a big molecule of RNA. It will bind to the promoter area of a gene. So it won't just randomly bind to any part of the gene. It has to be the promoter area, which is basically the area where the whole process of transcription is going to start. First off, again, you have to split the double helix, okay? And then you, by breaking those hydrogen bonds between the bases, you unwind it, and then you start the transcription process. Excuse me. You want to be familiar on how transcription can occur. So for example, if you were given a DNA strand, say of A, T, G, how would you transcribe that into a messenger RNA? So A would be transcribed as U, T would be transcribed as A, G would be transcribed as C. So that would be your messenger RNA that was transcribed out of that piece of DNA. Now these, that messenger RNA again, remember we make both the introns and exons. We have to get rid of the introns because those are really things that do not really code for proteins. So you want to eventually get rid of it, and we do that through these spliceosomes that we talked about. And now that mature messenger RNA can leave the nucleus through the nuclear pores. So there's really nothing new that we're talking about. This is just kind of like reinforcing the whole idea. We looked at it through the images. Now we are just reading how the process works. Now the second stage is translation, where the messenger RNA goes from the nucleus through the nuclear pore into the cytosol. It binds to a ribosome, which is your ribosomal RNA. And then the transfer RNA will read the codes on the messenger RNA. The transfer RNA, remember, has the anticodons, while the messenger RNA has the codons. The transfer RNA brings one amino acid at a time, and it runs down the messenger RNA, reading it. It starts at the start codon, and it keeps on moving down until it reaches one of the stop codons. Some cases we make a bigger protein than needed, or we have to activate the protein by splitting it into smaller portions, and that process is known as post-translational processing or post-translational splitting. Now, what is a mutation? A mutation is a structural change that happened to the DNA bases. So those 4 bases again, C, G, A, T, if one of them changes, that means that you have changed the sequence of the bases on your DNA. You've changed the manual, you changed the instructions, which technically means that now you are going to make a different kind of protein. Okay, so remember, the protein starts off as the sequence of bases that are found in the DNA. If you change that sequence, you have now changed the protein that needs to be made, or that is going to be made. A lot of times, though, mutations are silent. Okay, we call that a silent mutation, where it does not affect the protein being made. Okay, a lot of times, though, the mutation can lead to different diseases. It can also be deadly because you've made a protein which could impair the cell function or actually lead to cell death. So let me explain why some of them are silent mutations. And this takes us back to the redundancy. So if a mutation happened where let's do this one, where the A is replaced with a G, okay, we are still coding for the exact same amino acid. So your end result has not changed. So although there is a mutation that changed the base, it did not code for a different amino acid. Okay, and so we could have these silent mutations. We can live and die without even knowing that they were there. So the silent mutation is due to the redundancy in the codes. Now, how do we get rid of proteins? So we are going to be making lots of enzymes. Enzymes are proteins. We also make protein hormones, and we obviously cannot keep them forever, so they do eventually age off, and we do need to get rid of them. And that process is known as degradation. The simpler the structure of the protein, the easier it is to get rid of. So remember when we talked about protein, we said that there is a primary structure, secondary, tertiary, and quaternary structure to it. The more complicated the structure is, the harder it is to get rid of. Okay, so a denatured protein, which is an unfolded protein, okay, is easy to get rid of. It's easy to degrade that and break it down into its building blocks of amino acids. Know that the body will recycle those amino acids to make fresh protein. So how is it that the body knows that these proteins in particular are old and we need to get rid of them or degrade them? It adds a red flag, and that red flag is known as ubiquitin. And I kind of like to think of it as a red flag that says, you know what, protein, ubiquitin being a protein, okay? And that again is your red flag so that a whole bunch of enzymes known as proteosomes will come in and degrade the protein, break it down into its, break down peptide chains, break down the peptide bonds, and degrade it into its building blocks of amino acids. Again, the body is a very conservative thing. It won't get rid of those amino acids. It will just recycle them into making fresher proteins. Now this ends this chapter. Please let me know if you have any questions.